Namaste to all. This is Dr. Srinath Naganathan, Assistant Professor in Biotechnology. In today's session, we are going to see about RNA as genetic material and the different structures of RNA. Let's see. So RNA types and functions, if you see, it carries the genetic information from the DNA to protein biosynthetic machinery of the ribosome. So during the transcription process from the DNA, it is getting transcribed to form as RNA. And this process is actually called as heterocatalysis. So found both in the nucleus and in the cytoplasm. And the single-stranded RNA tends to assume a right-handed helical conformation dominated by base stacking interactions. So the thermodynamic stability of the RNA, if you see, it is the primary structure of the RNA refers to the sequence of the nucleotides. That means from the DNA, if it is adenine, it is going to be uracil in RNA. Thymine, it is going to be adenine. Guanine, it is going to be cytosine. And cytosine, it is going to be guanine. And the secondary structure in RNA is dominated by the Watson-Crick pairing, as it is observed in the bDNA conformation. So this interaction between the bases lead to the formation of the double helical structures of varying length, because as I said, the stem loop formation and other hairpin formation and other formations takes place in the RNA, single-stranded RNA, because they have the complementarity nature. So what happens is they join together to form a double helical structure. So the double helix adopt the A form, and the RNA secondary structure is generally more stable than its tertiary structure. So the formation of the secondary structure dominates the process of RNA folding, and the tertiary structure is formed by weak interaction between preformed secondary elements. Let's see the structures, how it is formed. So if you see the trans-esterification process taking place in the RNA, as I have shown here in this diagram. So in this, what happens is, it is actually the esterification process which is being involved here. So the first process is actually you can see that we are having two bases which are present here, okay, and it is linked by the 3' to 5', forming a phosphodiester form. By the trans-esterification process, due to the affinity, since it is a ribose sugar, it is the 2' position, it is a OH group is present. So due to the affinity which is present here, they form another bond formation, due to which the ester bond is formed between the 2' and 3' OH, 3' group. So the both are linked, forming the phosphodiester bond in the same base, and the other base is being removed. So after this, if they want to again undergo for the base pairing, so what they do is they go for the hydrolysis, in which the OH group is added, and thereby what happens is the OH group get attached to this 2' position, and the phosphate group is now released. Thereby the OH in the phosphate group which is remaining, it is free to form the phosphodiester bond formation with another base in the 5' position. This is how the RNA, it is actually maintained, or it is being stable in the secondary structure. So let's see the tertiary structure formation. So RNA duplexes are more stable than the DNA duplexes. So in the neutral pH, RNA dissolution requires 20 degrees Celsius more temperature than for the DNA. And the stability of the RNA-DNA hybrid is intermediate that of RNA and DNA. So the pseudoknot formation which can observe in maximum in RNAs, that is found in ribozymes and viruses. So you can see the structure, that the tertiary structure formation, it is because of the complementarity natures between the nucleotides, So what happens is, since it is forming the complementarity nature, it is actually being forming the tertiary structure. So let's see the types of RNA. So we are having totally 8 different types of RNA which are observed in eukaryotes and some in prokaryotes as well. So they are mRNA, tRNA, rRNA, snRNA, snORNA, tmRNA, XIST, and telomerase RNA. Let's see each in detail. So if you see mRNA, it encodes a chemical blueprint of our protein product. That is, directly from the DNA, the template strand, it forms the RNA, which is the mRNA, which is the blueprint, and which is used for the protein synthesis. So the DNA by transcription becomes as mRNA, and by translation goes for carries to the site of the protein synthesis by ribosomes. So in mRNA, as in the DNA, genetic information is encoded in the sequence of nucleotides arranged into codons consisting of 3 bases each. So monocistronic, that is code for a single protein, and polycistronic means code for many proteins. So tRNA, if you see here, as I said, they are having primary structure, which is a single, which is being formed during the transcription process. Secondary structure, that is a clover leaf structure, which is commonly available. Tertiary structure, all the tRNA have similar L-strand 3D structure, allows them to fit into P and A site of the ribosome during the translation process. The acceptor stem contains non-Watson-Crick base pairing, that is 7 base pair stem, that is base pairing by 5' terminal nucleotide with 3' terminal nucleotide, contains CCA at the 3' position and 5' having the OH end. And CCA 3', that is cytosine and adenine, which is present here. It is used for the recognition of tRNA by enzymes, and during the activation amino acylation process, it is critical in the translation process. D-loop formation, that is the D-loop, when it is being formed, it has 4 base stem ending in loop. So it has the dihydrouridine, and anticodon arm, it is for that is a recognition of at the codon in the during the translation process. That is a 5 base pair stem with loop having the anticodon. And T arm, that is the 5 base pair stem with T, psi, and C sequence. That is, psi is basically for the pseudo uridine here. Let's see the structures. So as I said, this is the clover leaf structures. So it is having the CCA, which is at the free end, and OH, which is the 3', and the phosphate at the 5'. And this is for the acceptor stem. This is actually the acceptor stem at which the amino acids can attach and can go for the recognition in the anticodon regions with the codons of the mRNA. And this is basically, if I is present in this RNA, it can go for the Wobble hypothesis, which we'll be discussing in later stages in the same topics. So this is a variable group which is formed here, and this is our T, psi, and C. That is the T pseudo uridine and that is the cytosine loop, and this is the D loop which is present. So miRNA. So this is the post-transcriptional regulators basically. So what they do is they bind the complementarity sequence on the target mRNA, and they results the translational repression plus gene silencing. So the size is 21 to 22 nucleotides, and they're derived from single-stranded precursor RNAs and fold back on themselves. So the double-stranded RNA with stem loop at one end is formed. So this is our pre-mRNA which is being formed. So siRNA, these are the small or the short interfering RNAs. That is why the name is short interfering. That is why SI. Sometimes it is also called a silencing RNA. And during the RNA interference for blocking or for removing some for the genetic analysis as well as for the treatment purpose gene therapies, the siRNAs are being used. So here the 20 to 25 nucleotides are present, which are double-stranded RNAs, and the role is RNA interference pathway, in which it is used commonly nowadays in the plant biotechnology, molecular plant breeding, in various gene therapies it is used. So it interferes with the expression of a specific gene here. So what happens is they form the short 21 nucleotides and the double-stranded RNA with two nucleotides, which is having the 3' overhangs on either side. These duplexes are produced by dicers, okay, and the first strand, it directs the silencing of the targeting mRNA called as the guide RNA, and the second strand is destroyed by the passenger strand, and it may be exo or endo-si RNA depending on the source of the RNA. So basically, if I want to create for any gene therapy for any particular disease, this is a basic method in which the siRNA plays a very vital role. So let's see how it is being taken place with the Psi and the miRNA. So if you see the first strand, it is the guide RNA, which is in red color, and the second, it is the passenger strand. So what happens in this particular Psi is it undergoes a cleavage at a particular site by the dicer, and the dicer, it is like a twisting mechanism. So here it identifies, make a cleave, it is twisting, and it is broken down. So once it is broken down, the passenger strand is removed, and now what you can do is the seed region is present, and this seed region you can go with the target strand, and target strand which is there, it can be present in the mRNA. So the mRNA can be identified by this seed region, and they can undergo a cleavage, and thereby the mRNA which is having the default or the defective gene for the protein can be removed, or it can be silenced by the another mechanism. So the similar mechanism you can observe in the miRNA. So in the miRNA, what they do is they form a knot-like system here. So this knot, what happens is they undergo the unwinding to repair it. So what happens is once it is unwinded, the below strand which is there, it acts as a seed region. And this seed region, when it is targeted to the mRNA, it can block or silence the particular reaction or particular protein formation, and thereby it can protect. So both the miRNA and the siRNA, they are actually used for the gene therapies, plant biotechnology experiments, for making new varieties of disease-resistant plants or disease-resistant or insecticide-resistant. So many different kinds of plants are generated right now using this particular mechanism. Let's see the piRNA. So the germ cells of the animals encodes the PIWI-PV. So this is actually the P element induced wimpy testis interacting RNAs. So this is suppresses the movement of the transposable elements in the germline. Transposable elements are, they are like jumping genes. They can move from one place to another. So these are actually helping like that. And the longer, that is 24 to 32 nucleotides are present in this, and the proteins which are there for successful gamete formation in mammals, and the roles is poorly understood till now. So still the research is going on in this particular subject, and there are so many other research which are going on different places related to this, in different studies related to animal biotechnology, related to human science, as well as in medical science. So let's see about XIST. So if you see the XIST, it is called as X inactivated specific transcript. So this RNA on the X chromosome, basically in the X chromosome, which is present in male and female as well. So it is basically the RNA gene on the X chromosome. The X inactivation process occurs because of this, because there is a particular site that's called as psych X chromosome inactivation center. So at this, what happens is the 17 kb in humans, it is present inactive chromosome and not on the active one. So those chromosomes which are inactive, in that it is present, and it is found on the placental mammals. Okay, so placental mammals means during the process of reproduction, the placenta is being developed. So at that time, it is being found. So X chromosomes lacking the XIST will not be inactivated, while duplication of the XIXT gene on the another chromosome causes inactivation of the chromosome also. So mechanism, how it works. So mechanism of the RNA interference is being done. So the double stranded RNA and RNAi pathway. So the role is regulation of the XIST promoters. So the dicer, actually the RNAi enzyme, what they do is they cleave this particular duplex of XIST at the beginning of the X inactivation to small 30 nucleotides, and thereby the XiRNA they repress the XIST on the probable active X chromosome. Let's see the telomerase RNA. So this is actually called as TERC, basically the telomerase RNA. So found in eukaryotes, and the component of telomerase used to extend the telomeres, and differ greatly in sequence and structure between the vertebrates, ciliates, and yeasts, but share a 5' pseudoknot structure close to the template sequence. The pseudoknot, that is a nucleic acid secondary structure, least two stem loop structures, that is half of one stem is intercalated between the two halves of the another stem. So in the process of mitotic division as well as the meiotic division, this is widely used for the telomerase formation at the end of the chromosomes. So this is present at the end region basically. So I hope one more is there. 1 minute, sorry. So that is snRNA, that is called as small nucleolar RNA. It is a class of small RNAs and guide chemical modification of other RNAs, mainly rRNA, tRNA, and small nucleolar RNAs. So we can just see the structures, how it is taking place. So what it does is it forms the Hoxtine base pairing. That is the Hoxtine base pairing. You can see that widely the normal pairing is different. This is the Hoxtine pairing, in which this is not the normal your adenine or guanine which is there, which is pairing. So it is having a difference. The next form is your cruciform DNA formation. So the cruciform DNA, this is your linear DNA. So in this linear DNA, because of the complementarity nature which is present, they can form a loop here, they can form a loop here. Based on this looping is creating the cruciform DNA formation. The other formation in this is the hairpin loop formation. So the hairpin loop formation, what happens is, here you can find that normally there is a since complementarity is there, so they form a hairpin structures, and this is normally present because of the particular condition. The most important one in this is the Tobacco Mosaic Virus, in which we will see that how the particular RNA is present and how it works. And it is the most serious pathogens causing the mosaic on the tobacco leaves, that is like brittleness, or you can say like mosaic, like brownishness on the structures, and thereby the leaf gets destroyed and it cannot be used further. So it is transmitted by the artificial inoculation, but not by insect vectors. So the most resistant virus, which can withstand the thermal death, almost death point of 90 degrees Celsius for 10 minutes. And this first virus that was crystallized in 1935 by W.M. Stamley in USA. So this is the structure of the Tobacco Mosaic Virus. And if you see the virus, it is looking like it is a protein capsid coat, okay, which is having the capsomeres. Okay, one are called as capsomeres. The central axis is almost like 40 Armstrong, okay, and the capsomeres is almost like 50 Armstrong. So the total is almost like 80 Armstrongs, almost like 180 is the total Armstrong distance of this, the circumference, or you can say like the diameter of the particular Tobacco Mosaic Virus. And the central, it is like a spring-like structures of the nucleic acid, that is the RNA which is present. So symptoms, what it does, it causes the Tobacco Mosaic Virus, it damages the solanaceous plants basically, that however infect the other plants too. So the several strains of the Tobacco Mosaic Virus has been reported. So the after infection, it develops the system of lighting of the leaf color, that is almost like greenish to brownish, almost like it becomes brittle and sometimes among the veins in the early stages. So what happens is the leaf of the tobacco, it causes the mosaic like structures. So half green, half brown, yellowish, some brittle like structure. So that is why what happens is those leaf cannot be used further. So this is what is actually happening. So here what happens is this is the uninfected leaf. And if you see, once the tobacco mosaic virus has been infected, the color actually becomes almost like yellowish and then becomes brownish and then it becomes brittle. So thereafter, it turns to a light and dark green mosaic symptoms. And among the veins, green color turns into dark green and terminal region turns into chlorotic. That is almost like whitishness, brownishness. Sometime dark green blisters appear in the leaf blades. And if the plants are infected early in the season, they become shunted. Sometimes what happens is they actually fall down. So the farmers face a lots and lots of problem because of this tobacco mosaic virus. So the from one plant to another plant of the tobacco, the symptoms varies, and the virus actually reduces the yield as the quality of the products. That is, the nicotine content is decreased by 20 to 30%. So structure wise, if you see, it was actually given by Franklin et al. In 1957 and described the structure of the tobacco mosaic virus. It is a rod shaped helical virus, which is measuring about 280 by 150 micron with a molecular weight of 39 into 10 to the power of 6 daltons. The virion is made up of 2130 protein subunits of identical size. Virion, what I am saying is the capsomeres, that is the structures which are there. It is almost like 2130 protein subunits of the identical size. And the protein subunits are arranged around the central hole of 4 nanometers or 40 Armstrongs. And each protein subunit is made up of a single polypeptide chain, which possesses almost like 158 amino acids. The molecular weight for this is 17,500 daltons. So inside the protein capsid, there is a single stranded RNA which is present, and it's a supercoiled, spirally coiled form of the helix in the right-handed direction. And the virus consists of 6,500 nucleotides, and in one turn, it has 49 nucleotides. Okay? And the number of protein subunits counting in 3 turns is 49. So 49 by 3 subunits per turn. And therefore, a single protein subunit is linked with 3 nucleotides of RNA. And the arrangements of the capsomere, it is as shown in the figure, you can see here. So it is like a leaf-like structures in a circular direction. So it starts from here and then turns like turn-wise it goes on. So that is why it is looking like a model. So this is how the arrangement takes place in the tobacco mosaic virus. And since, as I explained, the RNAs are actually having the 49 nucleotides per turn, and it is almost like 6,500 nucleotides which are in the right-handed direction. So with this, I hope that you got some idea about the RNases which are present and the tobacco mosaic viruses. So I hope that this gives you a better idea to understand about RNases and how it can be taken up for the transcription and translation processes. So let's see in detail all the other topics, like how the organization of prokaryote and eukaryotic genome is present and how it functions. Thank you, and hope that to see you in the next session with interesting topics. Thank you.